All right, first of all, quick review. Talking about viruses. First question. Where do viruses fit in the classification system? Like what kingdom do they belong to? They don't. Because they're not considered to be what? Living. There's a whole separate classification system for viruses. They're not in the normal system of taxonomy. Remember that the word comes from a Latin word meaning poison. They're obligate intracellular parasites. They have to be inside a cell to do something, and when they get in there, they cause damage. What's a bacteriophage? There you go. It's a way to interrupt Jake there. It's a virus. Jake interrupted Jake. No, it's a virus. You just wanted to keep it from looking bad, right? It's a virus that affects bacteria. It literally means what? What does it literally mean, the word? There you go. Huh? What's the word? What does it mean? Bacteria, what? Eater. Eat. Phage. Also remember that viruses have a protein coat called the capsid on the outside, and then either DNA or RNA, but not both, in the middle. It's called their nucleic acid core. We talked a lot about the lytic cycle. Before we do those, look at these pictures. Everything up there in blue is protein. Everything in red is DNA. And that's about what a virus is made out of. Whoops, skipped a couple there. Here are a couple more. Here's the flu virus, the influenza virus. Notice it's an RNA virus, number one, not DNA. And it has, in addition to that capsid, it's got an envelope on the outside, more of a protective coating. Which is more dangerous, DNA virus, RNA virus? How come? They mutate. They mutate really, really often. And here's the lytic cycle. Remember, the lytic cycle is the life cycle of a virus. First of all, it has to get into a cell. That's called attachment. Usually binds to a receptor for something else. And what's the trigger when something connects to a receptor? What's supposed to happen? If something connects to a receptor on your cell, it's supposed to pull it in. So the next thing that happens is penetration. The DNA or RNA, whichever one it may be, gets injected into the cell. Step 3, we're going to skip this lysogenic stuff for a minute. Step 3, your cells start doing whatever the DNA says to do, the virus DNA, which is usually make more viruses. And those get put together in this 4-step called maturation. And then finally, the virus is basically burst out of the cell, and that's what we call a release. That's the lytic cycle. Viruses make you sick because they kill your cells. They burst them. Now, there are a few viruses that can drop down into this other stage, lysogenic. What goes on there? They're just hanging out. Carter, we'll get that later. Don't worry about it. Their DNA becomes part of the host cell's DNA. And basically they're just sitting there waiting for an opportunity to go back into the lytic cycle. This is sort of like a virus in hibernation. It's there, it's not doing anything. Every time its host cell gets copied, so does it. The piece of viral DNA that's integrated into the host DNA is called the prophage, which literally means what? Prophage, what does it mean? Come on in. Before the eating. Because when it comes out of this lysogenic stage, it's going to go straight into the lytic stage, and it's going to start destroying it again. What did we say are 2 human viruses that seem to do that? 2 human viruses that seem to do this. Herpes. Yeah. Say again. Yes. Chickenpox, shingles virus. It's the same thing, right? You get it as chickenpox, it comes back as shingles. Many, many years later, and it can come back several times. All right, another thing that we want to mention are those things we talked about called retroviruses. What are retroviruses? What kind of viruses? RNA. Remember that they're exceptions to our major rule of genetics, the central dogma of genetics. This is what we call the central dogma. We talked about this last time. The process of going from DNA to mRNA is called what? Transcription. mRNA to protein is translation, and then proteins determine your traits. Every living thing that we know of does this. This is one of those central conserved properties. But retroviruses are an exception because they don't have what? They don't have any DNA. They just have RNA. So when they inject their RNA into a host, there's an extra step involved. They got an add-on to the central dogma. And that's called reverse transcription. Not only do they inject their RNA into the host, they also inject an enzyme called what? Reverse transcriptase. And it takes their RNA and uses it to make DNA. And that's kind of the way retroviruses work. Give me 4 examples of retroviruses. HIV. Now, let's put them up here. HIV, influenza, the flu, common cold or the rhinovirus, and Ebola. All those things are really hard to what? Yeah, well, common cold's not so hard to get rid of, but hard to prevent, right? Hard to keep you from getting them because what do all the time? They mutate. Can we get everybody's name on the list? Right. No. All right, let me back up a little bit. All right, there's a list of common viral diseases. What's the typical treatment for a virus? Nothing. Go home and rest, drink some fluids, hope your immune system is working right. There are some antiviral drugs. We talked last time that a lot of those AIDS drugs, they're inhibitors for certain virus enzymes, like this one, for example. A lot of the AIDS drugs target this enzyme. Because if it can't do reverse transcription, then what's never going to happen? If it can't take its RNA and make DNA, can it really cause you any problems? No. So if you block that enzyme, you can block the virus. They're developing more and more of these antivirals. There's this drug called Tamiflu, which is supposed to help you get over the flu easier. There's several drugs. They're not 100% ready yet, but for HIV, they're pretty good. That's the main one that they have the drugs for. Mentioned a couple other just pieces of information. There are these little things called viroids, which are sort of like viruses. But they're simply pieces of RNA, no protein coat. They're just single pieces of RNA. And as far as we know, animals, our immune system is sophisticated enough that they can't really make us sick. Our immune system will wipe those out. But they cause a lot of plant disorders. Plants get sick too. There are lots of plant diseases caused by these little infectious RNA segments. And then these guys right here are scary, prions. Anybody know what is caused by a prion? Something kind of dangerous. What'd you say? Mad cow disease. Mad cow disease, yeah. You've heard of mad cow disease, right? How do you get mad cow disease? Eat meat that's infected with this thing, this prion. Does it matter if it's cooked or not? Heat doesn't kill it. Because it's not alive. It's just a protein. And it's a protein that can self-replicate itself and cause chaos in your brain if you get in there. As far as we know, prions cause mostly degenerative disorders of the nervous system, like mad cow disease. There is a really strange one you might have heard of before called kuru. Anyone ever heard of that? This was discovered in Africa In what group of people? Wow. Y'all are quick this morning. Cannibals. Cannibals. There's still cannibals in Africa. And these particular cannibals, when did they eat somebody? No. They ate their enemies. And why did they eat them? It's like the ultimate insult. I beat you, I killed you, I ate you. Okay? It was like an insult. They weren't eating them just to be eating them. They were eating them to denigrate them. So what they found is that these cannibals, a lot of them kept getting this brain disorder like mad cow disease. And basically their brain just degenerated, and it's like Alzheimer's disease on steroids, sort of. Really fast, really bad degeneration of the brain. And what the scientists who were studying this group of people eventually figured out, it'd be kind of scary, scientists studying cannibals, right? Kind of thing. But regardless, they figured out they were getting this disease, they were transmitting it, because what's the main part of the body they were eating of these people? Their brains. They're eating their brains. And what they eventually did, at least this one group that they were studying, they eventually convinced them that by eating these brains, you are killing yourself. And I think that particular group, that this kuru was happening in, they convinced not to do it anymore. Whatever. Yeah, the enemy wins in the end, right? Eat my brain, I still get you kind of thing. But there's a lot of diseases that may be caused by these prions that we don't know about. There's some people that think Alzheimer's might be related to these things, that it might actually be catchable, which is kind of scary. Transmittable. Mad cow is another one. There are diseases like this in just about every mammal that we know of. There's a similar one in deer. There's a similar one in sheep. These really bad degenerative disorders of the brain. And the scary part is these things aren't alive. They're not heat sensitive, so cooking it won't usually get rid of them. Source here. All right, these terms over here, we've already talked about. Pathogen is what? Anything that can make you sick. Not just virus or bacteria. In humans, what are 2-3 other things that can make you sick? Fungi, protists, and one more. Worms. There's a lot of human pathogens. Probably some other things. We know an epidemic is a local outbreak of disease. And a pandemic, pan means what? Worldwide. I'm not sure that we talked about this last time. What does trans mean? Yeah, but what does just the root trans mean? It means between or across. In this case, it's when DNA moves from a virus into its host. It actually can put new DNA into its host genes. And that's what transduction is. It's when a virus transmits DNA to its host, new DNA. There are some scientists who think that a lot of the genes that we have today as humans were transmitted to us not through monkeys or whatever, but through viruses. That we actually got new DNA injected into us by a virus. A DNA is sort of like a syringe that shoots DNA into you. And part of that DNA becomes part of your own DNA. Kind of cool. All right, the other thing that we're going to talk about today are bacteria. So we'll come back to viruses over and over again. But we're going to talk about bacteria a little bit today. There are two main groups of bacteria. Eubacteria, which are the ones we're going to talk about mostly today. What's the other group? Archaebacteria, or archaea. We're not going to talk too much about them today. There's some important things to know about bacteria. They are prokaryotic. And again, if you break that word apart, prokaryotic, literally, what's the meaning of it? Before the nucleus, because they don't have a nucleus. We know that they do have cell walls. And what's the main function of those cell walls? What's the main function of pretty much any cell wall? To keep it from bursting when it's where?
Turgor pressure.
Yeah, when it's in water. And the turgor pressure is pushing on it. Their cell walls are made of a sugary protein compound called peptidoglycan. Sugar, protein. We know they don't have any membrane-bound organelles. Well, let's add that there. No membrane-bound organelles. Because they do have ribosomes. Everything has to have ribosomes because they're the parts that do what? Make proteins. Nothing can live without proteins. Gotta have proteins. They also don't have a membrane-bound nucleus. But they do have DNA. Their DNA works basically the same way yours does. It's the same chemical. It works the same way. It goes from DNA to mRNA to protein to trait. How many chromosomes do they have? One, and what's the shape? Circular. And it's just floating around where? It's just floating around in the cytoplasm. And it's called the nucleoid. That's just to let you know, hey, there's some DNA there, but it's not separated from everything else. It's not enclosed. A lot of bacteria are also surrounded by what's called the glycocalyx, or the capsule. That's on the outside. That's outside the wall. And mostly it's a mucus-like coating. Why would bacteria need to be surrounded by a mucus-like coating? I think we talked about this before. Maybe to be sticky, to stick to a surface, to move, like slide. What else? Yeah, to help it move better. But there's two other important functions that glycocalyx may serve. One of them is to protect against immune systems. It's like armor. And another is to protect against probably its worst enemy if you're a bacteria, which would be what? What would be the worst thing that you would need to worry about if you were a bacteria? Maybe there are viruses, that's not what I'm thinking about though. Drying out. Desiccation. No water. So it helps keep the water in. It's acting sort of like your skin. Water in. Now, another cool thing that some bacteria have are these little things called pili. P-I-L-I. Pili. They look sort of like cilia. They're little hair-like extensions. And they have two main functions. One of them is they help bacteria to stick to surfaces. They're sort of like bacteria Velcro. A couple of them stick to things. And the other one I'll get to in a second. I'm going to talk about these other two things here first. Bacteria have that one main circular chromosome. But in addition, they have some really, really, really tiny loops of DNA called plasmids. They just have a few genes on them. Very small loops of DNA called plasmids. This word is going to come up over and over again in this class because they're really important. So they're extra loops of DNA, tiny, tiny, tiny loops of DNA that bacteria typically have. The reason they're important, well, let me back up for a minute. How do bacteria normally reproduce? Asexually. And we call it what? Binary fission, or sometimes we'll call it prokaryotic fission. What's the good thing about that kind of reproduction? What's good about asexual reproduction? Make an exact copy. And what were you saying? You don't have to go find a mate. You don't have to put up with a mate, right? What else? Another benefit of asexual reproduction. It's fast. Really fast. How long does it take a bacteria one to become two? About 20 minutes. Depends on the species. Maybe about 20 minutes. It's fast. So that's a good thing. When would it make sense for bacteria to do asexual reproduction? When would it make sense for anything to do asexual reproduction? If we could do asexual reproduction, when would that make sense? It makes sense when the environment is pretty good and we're doing good in that environment, right? Everything's nice. Why not just keep making more, right? So when the environment is good, asexual reproduction is a good thing because your DNA seems to be working in that environment. The environment seems to be not changing. Makes sense to do asexual reproduction. When would it not be so good to do asexual reproduction? When things are changing. Because if you're asexually reproducing, what are you not doing very much of? Your DNA is not changing from generation to generation, which means you're not what? You're not evolving. Which means if the environment's changing, what's probably going to happen to you? You're probably going to die. You're going to go extinct. What's the purpose of life according to biologists? To make sure that what happens? That your DNA is passed on from your generation to the next, 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 to the next. It's the purpose of life according to biologists. To ensure that your DNA lives on forever. One way to do that is asexual reproduction, and that's great as long as the environment's going good. But when the environment's not working so good, you need a little evolution going on, right? You need to be able to change. And one of the main ways in biology we get that variation is through sexual reproduction. That's the purpose of sex, is to create genetic variation. Let's guys. Well, let me back up for a minute. There are some species of animals where there aren't any males. Right? And they usually make it okay until what comes along. No, I'm saying there aren't any males of that particular species. They make it okay until the environment starts changing. And then they start having problems because they can't what? Can't adapt fairly, can't evolve. Because their genes are all pretty much the same because one just grew into another. Other than bacteria, what else are we talking about here that can do this kind of thing? Asexual reproduction. Say again? Maybe. But we're talking about things more complicated than that. Like there's some salamanders, lizard kinds of things, some amphibians. If you watch Jurassic Park, they talked about this on there, right? That's true. There's lots of different animals that can reproduce this way. And they make it just fine until the environment becomes kind of weird. So what's the job of us guys? Why are we here, guys? When times get hard, what are we here to do? Save the world, right? That's what we're here for, guys. And we save the world by doing what? Making babies. Well, not just by making babies, but by making babies with what? Genes that are different. Because the whole idea of sexual reproduction is to take genes from two different organisms and mix them and change the gene pool, right? That's what sex is about, changing the gene pool, mixing genes, giving us more different kinds of genes in our group. And that protects us. If when that 1918 flu virus had come along, killed 30 million people in the world, if everybody had been the same genetically, what would have happened? Most of us probably would have died. A lot of people did die, right? 30 million people. But some lived because some were what? Some had genes that made them what? Maybe not either immune or more resistant to, right? They got over it easier. That's why we need variation. The more different we are genetically, the stronger we are. That's a problem for bacteria. Because the typical way they reproduce is one becomes another, becomes another, becomes another, becomes another, becomes another. But there's one thing that can help them. These little plasmids, they actually can swap like baseball cards. What will happen sometimes in bacteria, especially if the environment is going crazy, two little bacteria will get side by side and they'll hook up. That's kind of a bad choice of words, but okay. This is their main chromosome. They attach up. They attach up to each other using these little pili that we talked about. Think of them sort of like a straw. So they've attached themselves together with a straw. Pili. Remember, in addition to this one big chromosome that they normally have, they also have these little extra loops of DNA. What do we say these little extra loops are called? Plasmids. And when the environment gets bad, they'll attach together and they'll swap some of these. They'll switch. Not so much the fusion. This is an active kind of transport thing. But they swap pieces of DNA. That's sort of like what? That's sort of like bacterial sex. They're swapping genes. That's what sex is about, swapping genes. It's called conjugation.
All right, bacteria say something like that.
That's sort of what it is.
Okay.
So bacteria attach together, they swap plasmids, they swap DNA, and then they separate. Now, when they separate, it's still two bacteria, right? But it's considered sex. Because the two bacteria you have now are what? Different than the two that started with. They change the genes. That's what sex is about. Mixing up the genes so that we get variation, so that we're better able to adapt to a changing kind of environment. Does that make sense? All right. So conjugation is the act of two bacteria connecting together, usually via their pili, and exchanging plasmids. And that's a form of sex, a form of sexual reproduction. A way to give them genetic variation. All right, back. Wait on that. All right, bacteria typically come in three main kinds of shapes. Anybody know what they are?
Heart shape, spiral shape. One's like a circle, one's like a tail looking kind of.
Yeah, one's sort of like a capsule, a medicine capsule. Anybody know what we call that shape? That's the other one. This one's called a bacillus or bacilli. Bacillus. Shaped like a capsule. Another shape is like a sphere. And that one is called a coccus or cocci, if we're talking about plural. Sphere-shaped ball. And then what's the other one? Spirillum. It's spiral shaped, just like a corkscrew kind of thing. Spirillum. Just be familiar with those three shapes: bacillus, coccus, and spirillum. Now you've all heard of streptococcus, right? Bacteria causes strep throat and other things. Anybody have any idea what streptococcus means? It means a chain. So it's a chain of what? Coccus. So streptococcus, it's a bunch of these spheres all hooked together in a chain. You probably heard of staphylococcus, which causes what common problems? Staph infections. Staphylo means a cluster, like a cluster of grapes. So in staphylococcus, they're all clustered up like grapes. Maybe something like that. That's an inverted grape cluster, right? Get some of that out. All right, last couple of things we'll mention this morning. There's a lot of diseases in humans caused by bacteria. Strep throat. Tetanus. How do you get tetanus? All right, a lot of people think it's the rust that causes the tetanus. It has nothing to do with the rust. It's just that's where what like to live. The bacteria that cause tetanus like to live in dirty, dusty, rusty places. Has really nothing to do with the rust. That's just where these particular bacteria like to live. What does tetanus do to you? What's another name for tetanus? Sometimes it's called lockjaw. The bacteria cause your muscles to lock up on you. And it's not just the muscles in your jaw, it's muscles all over your body. It's important to be vaccinated against tetanus. Syphilis, common STD. What famous historical figure had syphilis? I don't know, I didn't know that, probably. Hitler had syphilis. There are some people, some historians who think syphilis saved the world. Because the beginning of World War II, the Germans were kicking the world's butt, right? I mean, literally. They were killing everybody. They wiped out all of Europe. We were having a hard time fighting them off. The Russians were having a hard time with them. And then all of a sudden, Hitler started making a lot of stupid decisions. Crazy, erratic kinds of things. What's one of the symptoms of late-term syphilis? It makes you start losing your mind. And a lot of people think that the reason Hitler was making those stupid decisions at the end that cost him the war is that syphilis was ravaging his brain. And if he hadn't have had syphilis, maybe that never would have happened. And the world might be a very different place. Hitler was kind of freaky with stuff, so I don't know that there's any proof that he did, but they were into like demon worship and crazy stuff. So I wouldn't doubt it. Pneumonia is also a bacterial disease. Pneumo, what does that mean? Pneumonia. Well, like if you have a tool that's pneumatic. What's a pneumatic tool? Runs out of air. Pneumo means air. Pneumonia obviously is an infection of your lungs, right? It has to do with the air. How do you treat a bacterial infection? Antibiotics. That's what antibiotics are for, to kill bacterial infections. Now, we hear about all these diseases that bacteria cause. And what's maybe the most famous bacterial disease, the one that's killed the most people, arguably? E. coli is a bacterial infection, but that's probably not salmonella. Salmonella's killed a lot of people, yeah. That's not what I'm thinking about still. The plague. The bubonic plague. The Black Death. Yeah, I see you. Is it still around? Yeah. There was a case in California not long ago. Why don't we get too concerned about that today? We have, no, we're not all into it. No, not all into it. We have antibiotics. And if the doctors catch what it is pretty quickly and they start giving you antibiotics, you're going to be okay. The thing is, if one of you turned up with the Black Death, your doctor at first is probably not going to have any idea what it is. Because how many of your doctors do you think has ever seen a case of the Black Death? Probably none of them. And they're not ever going to think about that being what it is. So it's probably not going to, until you start getting the sores, the buboes, and those kind of things, it's probably too late by that point in time. So it's still around. We don't get too beat up about it because we know we can treat it with antibiotics if we find out what it is, we diagnose it. Well, it's just like anything. Some people probably are resistant to it.
They're antibiotics for it.
Not always. We're not all resistant. And plus, it's been so long since the human population was exposed to it, we lost some of that resistance over the years, too. Somebody was back there asking a question?
Oh, that's there are antibiotics for it.
There are antibiotics that will kill it. All right, the last couple things here. even though there's all these bacterial diseases, by far, bacteria are more good than they are bad. They do a lot of important things in the environment, a lot of good things. Maybe their most important role is decomposition, what we might call nutrient recycling. If bacteria weren't around, we would be surrounded by piles of dead stuff, right? They break those materials down, and the nutrients either go back into the air or back into the ground. Nutrient recycling, that's really important. A lot of times rotting food stinks because of the waste products given off by bacteria during the process of eating waste.
Is the waste product what you're considering a recycled nutrient?
Partly. Sometimes those nutrients don't go back into the ground until the bacteria itself dies and it rots. But eventually those nutrients make it back to the ground. So that's hugely important. Another thing they do, and this is probably the last thing we'll mention here, is nitrogen fixation. How much nitrogen is on Earth?
A lot.
A whole lot. But where's most of it at?
Here.
In the air. 70% of the air is what gas? Nitrogen gas. But what's the formula for nitrogen gas? N2, right? And what holds those two N's together? Look at where it's at in the periodic table. It's in column 5. So what would two nitrogens have to do to connect up? What kind of bond do they have to form with each other to connect? Well, it's covalent, but what kind of covalent? What'd you say? A little more than that. Try again. They have to form a triple bond together, a triple covalent bond. So it looks like this. Turns out the more bonds there are, the harder that bond is to what? To break. And if we can't break that, can we use it? Turns out there are very few things on Earth that can use that nitrogen gas in the air. We breathe in that nitrogen, and then what do? Breathe it back out. We can't use it. Most things cannot use it. But nitrogen is really important because it's a main building block of what? Amino acids. It's also a building block of what else? It turns out to be really, really important. DNA. One of the building blocks of DNA they're called nucleotides, right? Nitrogen bases, A, T, G, C, right? You gotta have some nitrogen. Point being, bacteria are one of the few things that can take this into and break this bond and turn it into a useful form of nitrogen that other things can use. We'll talk more about that next time. That's probably a good place to stop.